Hi, my name is Alper Yurtseven. I'm a PhD student at Helmholtz Institute für Pharmazeutische Forschung in Saarland, Germany, in Professor Karina's group. Today, I will talk about how machine learning and phylogenetic analysis allow for predicting antibiotic resistance in Mycobacterium tuberculosis. Antimicrobial resistance is a global health concern that requires attention at the present time. Machine learning could provide valuable and accurate predictions of resistance-providing mutations. However, the phylogenetic relatedness of bacterial genome mutations reduces accuracy and can even lead to false predictions in traditional machine learning. Therefore, we have developed a pipeline that uses phylogenetic relatedness parallelism scores with machine learning to measure whether a certain feature is correlated with the population structure of a set of samples. We validated our approach using Mycobacterium tuberculosis genomes that were screened against 6 antibiotics. In our experiments, we rediscovered some known mutations as well as new and previously uncovered candidates. After the introduction, you can see the dataset we have used for our analysis. As you can see, we didn't have an equal number of resistant and susceptible strains, which presented our first challenge. On the right side, you can see a graphical representation of our pipeline, which I will briefly explain. First, we have collected the data from Petit database. Second, we have verified the data to eliminate bad reads and faulty config files. Next, we have performed variant calling against our reference genome, H37RV Mycobacterium genome, to collect variants. Using the variant output, we created binary mutation tables and filtered out some of the mutations. On the right side, you can see the filters we have applied on our data. At this point, we divided our research into 3 parts. The first part is genome-wide association studies, GWAS. The second part involves running machine learning on the data after the initial filtration. The third part is phylogenetic analysis and PRPS calculation. After calculating this score, we have filtered our mutations and removed 70% of the mutations highly related to phylogeny. We then trained the machine learning models with the same setup to demonstrate the effects of PRPS. Now, let's move on to the results. On the left side, you can see the GWAS results. The GWAS analysis employed linear mixed models with random effects to control for population structure. To identify important mutations in these results, we used Bonferroni p-value thresholds, calculated for each antibiotic separately. The red mutations here on the graphic represents those that were observed above the threshold. For phylogenetic analysis, we first created a phylogenetic tree using the panna cotta pipeline. On the right side, you can see the phylogenetic tree. We calculated PRPS on this tree and trained 3 models, one before phylogenetic analysis, one after phylogenetic analysis, and one where we randomly shuffled our data and selected 30% for training the machine learning models to demonstrate that our phylogenetic analysis is not random. As we can see, the top 30 PRPS scores have good method correlation coefficient MCC values, as do all the features, and they are much more better than the 30% random results. For machine learning, we have used 67% training and 33% test set approach. After training the models, we performed feature important analysis to determine which features, in this case mutations, were more important for the models. As a result, we observed previously reported mutations such as gyrase A mutation, which are known to confer fluoroquinolone resistance, as well as previously unreported variants like PAP-C or FTSH. For these important features, we use the structural classification tool, Structman, to classify their locations within the protein structure. On the left side, you can see an example visualization created by PyMol of the PAP-C and FTSH mutations. You can see the mutated amino acid in a red circle, both of which are classified as protein interactions. In conclusion, we have observed that applying phylogenetic analysis before training the machine learning model serves a valuable intermediate step for next experiments. Thank you.